Hello students, welcome to Crack NEET Biology. In the video, biotechnology and its applications NEET Previous questions discuss பண்ண போறோம். So இந்த chapterல இருந்து உங்களுக்கு average 4 to 5 questions கண்டிப்பா கேட்பாங்க. And NEET 2025ல இந்த chapterல இருந்து 4 questions கேட்டிருந்தாங்க. இந்த chapterோட detailed explanation videos பார்க்கணும் அப்படின்னா descriptionல videos link கொடுக்குறேன், ரெஃபர் பண்ணிக்கோங்க. So first biotechnological applications and agricultureல இருந்து கேட்டிருந்த questions பார்க்கலாம். First question, given below are two statements. Statement 1, transfer RNAs and ribosomal RNA do not interact with mRNA. This is wrong because translation process appo transfer RNA and ribosomal RNA, they directly interact with mRNA. So statement 1 wrong. Statement 2, RNA interference takes place in all eukaryotic organisms as a method of cellular defense. This statement is correct. RNA interference எல்லா eukaryotic organisms லையுமே natural defense mechanism நடக்கிற process. So statement 2 correct. So the answer is option B. Statement 1 is incorrect, but statement 2 is correct. Then silencing of specific mRNA is possible via RNA interference because of complementary double-stranded RNA. Option C. This question is very important. Nalla paathukonga. Then the Bt toxin in genetically engineered Bt cotton kills the pests by creating pores in the midgut. Option A. Then fourth question, steps involved in action of toxin in Bt cotton மாத்தி கொடுத்து correct sequenceல கேட்டிருக்காங்க. So first, Bacillus thuringiensis produce crystals with toxic insecticidal proteins. So இந்த toxin proteins bacteriaல இருக்கும்போது inactive prototoxins இருக்கும். Once insect இந்த Bt cotton ingest பண்ணுச்சு அப்படின்னா alkaline pH of the gut of the insect, it converts the inactive prototoxins to active form. So in the activated form of toxin, surface of the midgut cells bind பண்ணி it will create pores and cause the death of the insect. These steps involved in the action of toxin. So the correct sequence is first, Bacillus thuringiensis produce crystals with toxic insecticidal proteins. So in the toxin proteins bacteria இருக்கும்போது inactive prototoxins இருக்கும். So B, E. Then alkaline pH solubilizes the crystal. C. Then inactive toxin converted into active form due to alkaline pH of gut of insect. A. Then the activated toxin binds to the surface of the midgut cells, create pores and cause death of the insect. 5. So option D, B, E, C, A, D is the correct answer. Then given below are two statements. Statement 1, RNA interference takes place in all eukaryotic organisms as a method of cellular defense. This statement is correct. Statement 2, RNA interference involves the silencing of a specific mRNA due to a complementary single-stranded RNA molecule that binds and prevents translation of mRNA in கொடுத்திருக்காங்க. This is wrong because complementary double-stranded RNA molecule தான் வரும். So statement 2 is wrong. So option A, statement 1 is true, but statement 2 is false is the correct answer. Then given below are two statements. Statement 1, Bt toxins are insect group specific and coded by a gene Cry1AC. This statement is correct. Statement 2, Bt toxin exists as inactive prototoxin in Bacillus thuringiensis. However, after ingestion by the insect, the inactive prototoxin gets converted into active form due to acidic pH of the insect gut. This is wrong because alkaline pH la da adu activated form convert agum. So statement 2 is wrong. So option C, statement 1 is true, but statement 2 is false. Then transposons can be used during which one of the following? So gene silencing la the mobile genetic elements transposons use agum. Option B. Then Bt cotton variety that was developed by introduction of toxin gene of Bacillus thuringiensis is resistant to insect pests. Okay, option A. Then what triggers activation of prototoxin to active toxin of Bacillus thuringiensis in bollworm? So alkaline pH of the gut da, it triggers the activation of prototoxin to active toxin. Option D. So alkaline pH of gut nalla ஞாபகம் வச்சுக்கோங்க. Then which of the following is true for golden rice? So golden rice nale vitamin A enriched ஞாபகம் வச்சுக்கோங்க. So option B, it is vitamin A enriched with the gene from daffodil. Then 11th question, in RNAi the genes are silenced using complementary double-stranded RNA. Option A. Then 12th question, which part of the tobacco plant is infected by Meloidogyne incognita? So root of the tobacco plant da, it is infected by this nematode. Option B. Then Golden rice is a genetically modified crop plant where the incorporated gene is meant for the biosynthesis of vitamin A. So golden rice nale vitamin A nalla ஞாபகம் வச்சுக்கோங்க. Then in BT cotton, the BT toxin present in plant tissue as prototoxin is converted into active toxin due to the alkaline pH of the insect gut. This is a repetitive question, very important. Okay. Then 15th question, which of the following BT crops is being grown in India by the farmers? So option D, cotton is the correct answer. Then 16th question, RNA interference involves. So it involves silencing of the specific mRNA due to complementary RNA. Option B. Then consumption of which one of the following foods can prevent the type of blindness associated with vitamin A deficiency? So vitamin A nale golden rice. Option C. Then 18th question, the process of RNA interference has been used in the development of plants resistant to nematodes. Option A. So nematode resist பண்றதுக்காக தான் RNAi process யூஸ் பண்ணுவாங்க. Okay. Then tissue culture ல இருந்து கேட்டிருந்த questions பார்க்கலாம். The capacity to generate a whole plant from any cell of the plant is called totipotency. Option A. Then which of the following are fused in somatic hybridization involving two varieties of plants? So ரெண்டு varieties of plants ஓட protoplasm somatic hybridizationல fuse பண்ணுவாங்க. Option C, protoplasts. So protoplasmன்றது cells without cell wall. Okay. Then match list 1 with list 2. Protoplast fusion, potato plus tomato, P2. Then plant tissue culture, totipotency, Q1. Meristem culture, virus-free plants, R4. Micropropagation, soma clones, S3. So option C, P2, Q1, R4, S3 is the correct answer. Then a protoplast is a cell without cell wall, option B. Then fifth question, a technique of micropropagation is somatic embryogenesis, option D. Then to obtain virus-free healthy plants from a diseased one by tissue culture technique, which part or parts of the diseased plant will be taken? So both apical and axillary meristems will be taken to obtain virus-free healthy plants. Okay, option C. Then tissue culture technique can produce infinite number of new plants from a small parental tissue. The economic importance of the technique is in raising genetically uniform population identical to the original parent. Option A. Then which part would be the most suitable for raising virus-free plants for micropropagation? Meristem is the correct answer. Option C. Then biotechnological applications in medicine ல இருந்து கேட்டிருந்த questions பார்க்கலாம். Which of the following genetically engineered organisms was used by Eli Lilly to prepare human insulin? So human insulin produce பண்றதுக்கு Escherichia coliன்ற bacterium யூஸ் பண்ணாங்க. Option C. Then why can't insulin be given orally to diabetic patients? So orally insulin கொடுத்தா it will be digested in the gastrointestinal tract. Option D. Then match list 1 with list 2. Genetically engineered human insulin, E. coli, A2. Genetically modified cotton, GM cotton, Bacillus thuringiensis, B4. Then adenosine deaminase deficiency, gene therapy, C1. Then ELISA, antigen antibody interaction, D3. So option D, A2, B4, C1, D3 is the correct answer. Then again match list 1 with list 2. So gene therapy allows correction of a gene defect. A4. RNA interference, cellular defense. B3. ELISA, diagnostic test for AIDS. C2. Then gel electrophoresis, separation of DNA fragments. D1. So option C, A4, B3, C2, D1 is the correct answer. Then select the incorrect statement regarding chemical structure of insulin. So இன்சுலினோட கெமிக்கல் ஸ்ட்ரக்சர் பார்த்தோம்னா அது ப்ரோ ஹார்மோனா இருக்கும்போது the extra stretch of peptide chain C அதோட அட்டாச் ஆயிருக்கும் and இந்த ப்ரோ ஹார்மோன் மெச்சூர் ஆகும்போது the free C peptide it will be removed from the mature insulin. So A peptide and B peptide which is connected by means of disulfide bridges தான் mature insulin. Okay. Now coming back to the question, select incorrect statement regarding chemical structure of insulin. Option A, mature insulin molecule consists of 3 polypeptide chains A, B, C ன்னு கொடுத்திருக்காங்க. This is wrong. ஏன்னா mature insulin consists of only 2 polypeptide chains A and B மட்டும்தான். Option A is the incorrect statement. Whereas option B, insulin is synthesized as prohormone which contains extra stretch of C-peptide. This is correct. C-peptide is not present in mature insulin molecule, correct. Polypeptide chains A and B are linked by disulfide bridges. This is also correct. So option A the incorrect statement. Then, which one of the following techniques does not serve the purpose of early diagnosis of a disease for its early treatment? So, given options la paranga PCR, ELISA, recombinant DNA technology, இது எல்லாமே it serve the purpose of early diagnosis of a disease. Whereas serum and urine analysis, it does not serve the purpose of early diagnosis of a disease. So, option D is the correct answer. Then, in gene therapy of adenosine deaminase deficiency, the patient requires periodic infusion of genetically engineered lymphocytes because genetically engineered lymphocytes immortal cells கிடையாது. அதுக்கு ஒரு definite lifespan இருக்கு. அதனால periodic infusion of genetically engineered lymphocytes patientsக்கு தேவை. So option D, genetically engineered lymphocytes are not immortal cells is the correct answer. Then eighth question. Statements related to human insulin are given below. Which statements are correct about genetically engineered insulin? So genetically engineered insulin pathina correct statements கேட்டிருக்காங்க. Statement A, prohormone insulin contain extra stretch of C-peptide. இது genetically engineered insulin ஓட characteristic கிடையாது. So A கிடையாது. B, A peptide and B peptide chains of insulin were produced separately in E. coli, extracted and combined by creating disulfide bond between them. So this statement is correct with regard to genetically engineered insulin. Then insulin used for treating diabetes was extracted from cattle and pigs. So இது animal derived insulin ஓட characteristic, not genetically engineered. So C is wrong. Then D, prohormone insulin needs to be processed for converting into a mature and functional hormone. This is wrong. This is not genetically engineered insulin. Then some patients develop allergic reactions to the foreign insulin. This is also wrong. So genetically engineered insulin in the allergic reactions minimize பண்றதுக்காக தான் இத produce பண்றாங்க. So E wrong. So B only, option B is the correct answer. Then ninth question, when gene targeting involving gene amplification is attempted in an individual's tissue to treat disease, it is known as gene therapy, option C. Then 10th question, nowadays it is possible to detect the mutated gene causing cancer by allowing radioactive probe to hybridize its complementary DNA in a clone of cells, followed by its detection using autoradiography because, so இப்போ மியூட்டேட்டட் ஜீனா இருந்தது அப்படின்னா அதுக்கு ப்ரோபோட காம்ப்ளிமெண்டாரிட்டி இருக்காது, அதனால அது போட்டோகிராஃபிக் பிலிம்லயும் அப்பியர் ஆகாது. ஸோ அது மூலமா மியூட்டேட்டட் ஜீன் காசிங் கேன்சர் அதை டிடெக்ட் பண்ண முடியும் அப்படின்னு சொல்றாங்க. ஸோ ஆப்ஷன் டி, மியூட்டேட்டட் ஜீன் டஸ் நாட் அப்பியர் ஆன் போட்டோகிராஃபிக் பிலிம் as the probe has no complementarity with it. Option D is the correct answer. Then 11th question. For effective treatment of the disease, early diagnosis and understanding its pathophysiology is very important. Which of the following molecular diagnostic techniques is very useful for early detection? அப்படின்னு கேட்டிருக்காங்க. So given optionsல பாருங்க, hybridization, blotting இதெல்லாம் specific gene sequence or proteins identify பண்றதுக்கு யூஸ் பண்ற techniques. Whereas early detection of the diseaseக்கு ELISA technique யூஸ் பண்ணுவாங்க. Option D is the correct answer. Then 12th question, with regard to insulin, choose correct options. Statement A, C-peptide is not present in mature insulin. So this statement is correct. Then statement B, the insulin produced by rDNA technology has C-peptide. This is wrong. So rDNA technology மூலமா produce பண்ற insulinல C-peptide இருக்காது. So statement B is wrong. Then the pro-insulin has C-peptide. This statement is correct. Then statement D, A peptide and B peptide of insulin are interconnected by disulfide bridges. So this statement is also correct. So given options la pathina, option D, A, C, D only is the correct answer. Then the adenosine deaminase deficiency results into dysfunction of the immune system. Option B. Then match the following columns and select the correct option. BT cotton Bacillus thuringiensis. A4. Adenosine deaminase deficiency gene therapy. B1. RNAi cellular defense. C2. PCR reduction of HIV infection. D3. So option A, A4, B1, C2, D3 is the correct answer. Then 15th question, which of the following statements is not correct in கேட்டிருக்காங்க. In man, insulin is synthesized as a pro-insulin, correct? The pro-insulin has an extra peptide called C-peptide, correct? Genetically engineered insulin is produced in E. coli, correct? Whereas option C, the functional insulin has A and B chains linked together by hydrogen bonds and கொடுத்திருக்காங்க. This is wrong because they are linked together by disulfide bonds. So statement C தான் the incorrect statement. Then 16th question, what kind of therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency? So gene therapy was given, option A. Then the two polypeptides of human insulin are linked together by disulfide bridges, option B. Then the first human hormone produced by recombinant DNA technology is insulin, option A. Then 19th question, which one of the following vectors is used to replace the defective gene in gene therapy? So given options la pathina adenovirus, option A is the correct answer. So adenovirus, retrovirus, இதெல்லாம் the common used vectors for human applications. Okay. Then transgenic animals இருந்து கேட்டிருந்த questions பார்க்கலாம். Match list 1 with list 2. Alpha 1 antitrypsin, it is used to treat emphysema, A3. Then cry1 AB control corn borer, B4. Then cry1 AC control cotton bollworm. So cry2 ABU, it controls cotton bollworm. Okay. So C1. Then enzyme replacement therapy, ADA deficiency, D2. So option C, A3, B4, C1, D2 is the correct answer. Then maximum number of existing transgenic animals is of mice, option B. Then ethical issues learn the questions பார்க்கலாம். First question, given below are two statements. Statement 1, the Indian government has set up GEAC, that is Genetic Engineering Approval Committee, which will make decisions regarding the validity of GM research. Statement 1 is correct. Statement 2, biopiracy is the term used to refer to the use of bioresources by native people னு கொடுத்திருக்காங்க. This is wrong. So biopiracy அப்படின்றது use of bioresources by multinational companies and organization without authorization from the concerned country or its people. So statement 2, it's wrong. So option A, statement 1 is true, but statement 2 is false. Then a new variety of rice was patented by a foreign company. Two such varieties have been present in India for a long time. So this is related to basmati rice, option D. Then third question, in India, the organization responsible for assessing the safety of introducing genetically modified organisms for public use is Genetic Engineering Approval Committee, option D. In use of bioresources by multinational companies and organizations without authorization from the concerned country and its people is called biopiracy, option B. Then fifth question, which body of the government of India regulates GM research and safety of introducing GM organisms for public services? So Genetic Engineering Approval Committee தான் இதை regulate பண்ணுவாங்க, option A. So biotechnology and its applications இருந்து கேட்டிருந்த NEET Previous year questions பார்த்துட்டோம். இந்த வீடியோ உங்களுக்கு யூஸ்ஃபுல்லா இருந்தா என்னோட சேனல் சப்ஸ்கிரைப் பண்ணிக்கோங்க. இந்த வீடியோ பிடிச்சிருந்தா லைக் பண்ணுங்க, உங்க கமெண்ட்டை போஸ்ட் பண்ணுங்க. Thank you.